Hello everyone. In this video lecture, I will be discussing to you the continuation of amino acids and proteins. Okay, so we are now on the types of protein based on its solubility and its physical property. So we have three. The first one is the simple proteins. The second one is we have our compound, or we can term that one as conjugated proteins. And the third one class is we have the derived proteins. So let's start describing the simple protein. So, in a simple protein class, this is a type of protein in which only amino acid residues are present. So, very simple. There are no combination of any form of molecules. It's just the residue of the amino acid. So, they are the protein which, on complete hydrolysis, yield only amino acids. After the process of hydrolysis, so the use of water from the term hydrolysis. So we have covered this one on our first discussion. So the product of this process is only amino acids. Example of this type of protein is we have our globulin and also our albumin. The second one class is we have our conjugated protein. So this is a protein that is composed of one or more non-amino acid entities that can be present or is present in its structure in addition to one or more peptide chains present. So the amino portion of this conjugated protein class, this is called apoprotein. And the non-amino acid portion, which may be organic or inorganic, it is called a prosthetic group. When we say prosthetic group class, this is a non-amino acid group that is present on the conjugated protein. So basically, our conjugated protein is uh, one has one amino acid and an addition of non-amino acid. So the name of the conjugated protein is derived from the prosthetic group. So we have here an examples. So we have hemoprotein. So that is an example of a conjugated protein. And the prosthetic group is heme unit. So that is how we are going to name this type of protein. So another one is we have our lipoprotein. So the prosthetic group now is the lipid, which functions as a carrier of our lipid or fats in our body. Another one is we have an example here, our metalloproteins. So the prosthetic group is the metal ion. So its function class is to store iron. So we term that one as ferritin. So that is the storage form of our iron. And the rest of the examples that we have on the screen. And the third one is we have our derived protein. So derived proteins class, they are partial to um, complete hydrolysis from the simple or conjugated proteins. So by which the action of the acid class in this process involves enzymes or alkalis. So this one includes our primary derived protein, and also we have our secondary derived protein. So from the name itself, it is derived. So it was taken from our simple or conjugated by the process of physical or chemical attraction or conjugation. So we have examples here written on the screen. So we have our peptones, we have our fibrin that is responsible for clotting mechanism in our body, as well as our coagulated proteins. Now, on the molecular length and shape naman of our protein class. So, the first one is we have our fibrous proteins. So, example of this one is our collagen that can be found on the skin, keratin that can be found on our skin as well, on our nails, on our hair. So, we have elastins, myosins, and we have fibrin. They are tough, so since they functions as covering in general, they are insoluble and they compose of fibers and sheets. So this one class, so since we have myosins, it can be found as well on our muscle tissues. So they are elongated in shape with one dimension much longer than the others. The second one is we have our globular proteins. So this type of protein class, they are water soluble, and the example of this is our hemoglobin, we have our myoglobin, we have insulin, and also we have our immunoglobulins. So the length of this or width of this type of protein class, it is less than 10. So they are not that um lengthy or wide compared with our fibrous proteins. And the third one is we have the membrane protein. So this one class, it is the opposite of our globular. So di ba our globular, they are water soluble. For this type of protein naman class, they are water insoluble. So they can be found on the membrane system of a cell. And example of this is our receptor protein. So they cannot be found on the surface, no, since they are insoluble in nature. And the main purpose of this membrane proteins class is to allow cells to connect to each other. So they serve as a mode of communication, or they can be a mode of transportation. So to better understand our fibrous proteins and our globular proteins, so we have here a table that may compare these two structures of our protein. So according to its shape, so fibrous proteins, they are long and narrow, while our globular proteins naman, they are rounded and spherical. For its role, so fibrous, structural, covering, protection, for protection. While for our globular naman class, so they serves as a means of transportation. And solubility in water, our fibrous, they are insoluble, while our globular, they are soluble in water. So the sequence of amino acid, so fibrous proteins, repetitive amino acid, while our globular class, they are irregular ang composition. And for the stability, so our fibrous, they are less sensitive to changes in heat and pH, while our globular proteins naman, they are more sensitive to changes in heat and in pH. It's because of the irregular arrangement of their amino acid, and as well as class for its function. And so we have here listed our examples. So collagen and keratin for our fibrous, and hemoglobin, insulin, and catalase naman for our globular proteins. Okay. So, more examples. So kindly screenshot na lang this one for you to have a reference of the following examples as well as its function. Now, let's proceed with the physical and chemical properties of our proteins class. Okay, so this is the general characteristics. So generally, they are tasteless in nature. They are colorless. There are some maybe that can be with color, and but they're very few lang. And they are insoluble in fats, and also different ang kanilang pagka-soluble with water. There are some proteins that can be insoluble in water. When it comes naman to their molecular weight, so they have a high molecular weight, so they are heavy, naturally heavy in nature. So they can be modified as well when have this attraction with the pH, the UV, and the heat and organic solvents. So we will be covering that one later on. And the last one is they are very reactive and highly specific. Okay, so we have this term, they are amphoteric in nature. So what is amphoteric class? When we say amphoteric, they can act as an acid or base. So depending on the pH of the medium that they are referring with. So when we say acid class, again, the charge of the molecule is negative, and when we say base, the charge is positive. So we also learned, from our previous discussion that in neutral solution, our carboxyl groups, they have a tendency to lose protons. So they are releasing protons class. Therefore, they produce a negatively charged molecules. And for our amino groups naman class, they have the tendency to accept these protons. Therefore, by accepting protons, they produce a positively charged na mga molecules. So, um, this such molecule, they are termed as Zwitter ion. So when we say Zwitter ion, that is from or derived from a German term meaning double ion. Okay, so that is our that is the meaning of amphoteric. So it's in the neutral solution. So we have covered this one already, no? Those proteins that contains acidic groups. Okay, so we can see in their molecular formula the acidic groups. So they are negatively charged. So we have our aspartic acid, asparagine, glutamic acid, and our glutamine. Okay, so I have mentioned this one, no? Zwitter ion or double ions, or we can term this one as well as dipolar forms. Okay, so it is a molecule that has a positive charge on one atom and a negative charge on another atom, but which has no net charge. So Zwitter ion class, they can change when the pH of a solution contains amino acid, and it can be changed as well from neutral either to acidic or by adding or it can be they can be basic as well depending on the substance that we added on our molecule. So for acidic, it can be changed by adding hydrochloric acid, but if they will turn basic if we add sodium hydroxide. Okay, so as what I have mentioned earlier, in acidic solution, our sweeter ion, they form a positively charged ion, or they accept a proton to form a positively charged ion. While on a basic solution, our sweeter ion naman class, they loses a proton and a negatively charged molecule is formed. So let's just take note, class, that the net charge of our sweeter ion is 0. So it is only dependent on the solution that we added. Okay. Okay, so we have, for a while, we have an example here. So if the Zwitter ion is on the middle, our neutral solution. So it will depend on the added solution. It can be acid or it can be basic. So it will be dependent on the molecule or the solution that was added. Okay, so for acidic, positively charged molecule is added. And on the basic naman is negatively charged molecule is added. Okay, so more examples. We have more examples in here. So our proteins class, they have this isoelectric point. So when we say isoelectric point class, this is there is a pH present on our molecule, which there is an equal number of positive and negative charges present. So therefore, because of these charges, it produces no net charge situation. Okay, so when we say no net charge class, so the pH value now of our amino acid solution, it is called isoelectric point. Okay, so again, when we say isoelectric point, no net charge. Okay, so there is no presence, or sorry, there is equal number of positive and negative charges present. Okay, so we can measure the isoelectric point of an amino acid or of a protein by observing the behavior of an electric field. Okay, so if there is enough number of if there is enough number of positive and negative charges, of course we can see in the process that there will be no movement. Okay, so electrophoresis. So this is the process in which the charged molecule is attracted to the electrode of the opposite charge. So the principle of this process class is that it is the process of separating charged molecule on the basis of their migration toward charged electrodes associated with an electric field. So the process of electrophoresis class at a high pH, an amino acid has a negative charge and migrates towards the positive electrode. And on the other hand, at low pH naman, the amino acid, the opposite is true. With a net positive charge, the amino acid migrates now towards the negative electrode. Okay, high pH amino acid will charge to the positive electrode. At low pH, our amino acid will now migrate towards the negative electrode. So the isoelectric point of this process, it does not occur. It's because class Zwitter ions present have no net charge. So that is why there is no electrophoresis or there is no migration, since there is equal number of charges on each ends. So this is a machine that applies a principle of electrophoresis. So maybe you can perform this one in your laboratory. So there are times, class, that our proteins will denature. Okay, so what is this phenomenon? What can be the factor involved in the denaturation of proteins? So this is the partial or complete disorganization of a protein's characteristic three-dimensional shape as a result of disruption of its secondary, tertiary, and quaternary structural interaction. So the reason for this, or the factor for this, it can be the presence of high or low temperature, it can be high or low pH, it can be the presence of organic solvents, it can be because of the detergents added, or it can be because of the heavy metals attached on our protein molecules. So we will be covering this one by one. Okay, so the last one is our because of the mechanical stress. So it can be a factor as well, class, for our protein to denature. So just take note, class, during protein denaturation, so the primary structure of our protein is not affected since the process or the reaction of denaturation are not strong enough to break the peptide bonds. Okay, so therefore, our primary structure, which is the basic structure of our proteins, remains the same even after denaturation. Okay, so let's cover first the temperature. Okay, as the temperature increases, it also increases the rate of molecular movement within the solution, and the bonds within the protein begin to vibrate more. So, they coagulate when the temperature rises, it harden or it's stiffen. Okay, so it lose their characteristic, its three-dimensional conformation, and become completely disorganized. So this can be achieved plus if the temperature reached 56 to 60 degrees Celsius. So coagulation will occur as the protein molecules then unfold and become entangled. They have aggregated to become solid. So as what we have here in the illustration, no, from a like telephone wire, once it is applied, so the protein strand will start to denature, and if further heat will be applied to a denatured protein, it will result to a conjugated protein. So mas more ang loose ends on our protein strand. So for example, our microorganism. So that is why we are cooking our food, no, in order to kill the microorganism. So we are heating or we are sterilizing our kanang mga glasswares, our kitchenwares in order to remove, okay, the microorganisms that we have. Okay, so for example, our favorite na ham, our favorite na bacon, so they can have these parasites, no, that can result to, or they can have this Trichuris trichura that can result to trichinosis. So by cooking them, it can denature the parasite's protein and it can now be healthy for eating. Okay, another one is the pH. When the pH of a protein solution is above, is above the PL, all the protein molecules will have a net negative surface to charge. So below, so they have a net positive charge. So the protein molecules no longer have a net surface charge. As a result, they no longer strongly repel one another and are at their least soluble. So it will depend on the pH charges. So um, acidic it's negative, basic it's positive. The next one is our organic solvents. So they are polar organic solvents in nature proteins by disrupting hydrogen bonds within the protein. In addition to forming hydrogen bonds, the solvent example is water. So once water is added, it can disrupt our protein structure since it can interact with our hydrogen bonding. And we also have our detergents. So our detergents class, they affect our R group interaction. So, and then when interacts with proteins, they disrupt the hydrophobic interaction, causing the protein chain to unfold. And we have here the heavy metals, example, our mercury or lead. So they may form bonds with negatively charged side chain groups, and they can interfere with the salt bridges formed between amino acid. So they can remove the bonding by the presence of this mercury. So it can lead to denaturation as well of our protein. And then we have our mechanical stress. So steering, whipping, or shaking can disrupt the weak interaction that maintain protein conformation. Okay, so by shaking, no, by creating a movement, so especially for our weak na types of bonding, so it can remove or disrupt our or denature our protein. So here is an example of our denaturating agent that may cause changes or disruption on our proteins and its mode of action. So kindly, kindly screenshot so that you will have a better understanding about this. So more examples. Okay, so we have an agitation. Okay, so it can disrupt hydrogen bonding and hydrophobic interactions. Okay. And so the last type of protein is we have our immunoglobulins. So as you all know, our immunoglobulins, they can also be called as our antibodies and they are produced by our plasma cells. Plasma cells class, when you're going to centrifuge a blood, the liquid portion of your blood, so that is our plasma. That is where our immunoglobulins can be found. So they are highly specific and they has a memory. Okay, so they can recognize from self if this molecule is naturally occurring or this molecule is non-self, which is our antigen. So they contain 4 peptide chains that are connected by disulfide bonds and arranged in a Y-shaped quaternary structure. So all in all, plus we have 5 different types of immunoglobulins. We have the IgG, IgE, IgD, IgM, and IgA. So each of these types of immunoglobulins or antibodies class, they have different function. So in the table here, so we have here its function. Our IgA, so this can be found on our body fluids such as the mucus, saliva, tears, and breast milk. So its function is to protect us against pathogens such as our viruses and the bacteria. And our IgD, so they are part of our B cell receptor. This function is to activate our basophils and mast cells. So they activates our WBCs. Then we have our IgE. So its main function plus is to protect against parasitic worms. So other than protecting us from parasites, they are also responsible for allergic reactions. And then our IgG class, so this one is can be present on the placenta of the mother. So they can cross into the placenta of the mother. And we have IgM. So IgM, it is penta, or there are five Y nuclear bodies present on this type of immunoglobulin. So this is for the early stages of immunity. So they are the first to arrive, the first antibody to arrive once there is a foreign invasion or pathogen that entered our immune system. Okay, so that's it for our continuation on the properties, on the structure, on the kind or on the kind of the denaturing materials that may alter the composition of our proteins. Thank you everyone for listening and have a good day.